Okay, so I thought tonight's kind of a mishmash of a bunch of stuff. So I want to do another example because you weren't here, right? Okay, you weren't here on Monday. So I want to do another example of the DNA messenger RNA thing, just so you've got that down. And then we're going to go ahead and finish up the notes. And then I did not make up a review game for the exam. Rachel's very upset with me about that. But
no, I'm not.
No, I know what you're saying.
I like that you made that thing.
But I literally went through the exam, and I have got a review sheet that goes through everything you're going to need. So we'll get all of that done. Take a little bit of a break, go to lab, and we're not to the point. We're fortunate because we get to go to lab every week, whereas some of the other classes are every other week. So we're not to the point where we're doing anything new yet. We are still doing all the muscles. We're doing the origin, insertion, and actions, learning all the bones and the bone markings. So we will be in lab as long as anybody wants to be in lab. I mean, this is a great opportunity to review everything, make sure, since I goofed on the semimembranosus and semitendinosus, sorry about that. Make sure we go over that and we know that. Go over the bone markings again like we did. Make sure you're comfortable with all those in the bones. And then again, for the origin, insertion, and action, if you want to do them on the cadavers and on the skeletons, that's fine. If you are like me and you just want to memorize them like that sheet, that's fine too. So, we're kind of to the stage before the lab practical won't be next week yet. It'll still be the week after. So we still have one more week before the lab practical. So we've got lots of time. And like I say, I'm here till 10 if you want to be. So that the next couple weeks will pretty much be, that. How much do you need to look over? How much do you want to look over? That kind of stuff. So that's kind of the way the night's going to go. Bree, do you want?
Yeah, okay, I'm sorry.
No, it's okay.
So when we're looking at like the bones, when you're going to randomly pull out, that's not the action insertion.
No. There's Sandy. Hey, there she is. How are you? Good. Better?
Yes.
Good. No. The origin, insertion, and action is literally, I apparently reach into a bowl and I just pull out 5 of the muscles. That page I gave you?
Yeah.
I just pull out 5 random ones.
Okay.
And
then the bones one will just be around.
They'll be around the room. Yeah. So there will be bones around the room with cards in front of them. Some of them will say, name this bone. Some of them will say what bone marking is like at letter A or number 1 or what. They have little pieces of tape on them.
Okay.
And then there are 10. I think there are 10 like random multiple choice questions. And I will find out next week kind of what those are about. Whether they're about particular markings, whether they're I don't know exactly what the subject matter for the multiple choice are. But I'll make sure I find that out before next week so that there are no surprises. Okay? Shandy, I was just saying, tonight's kind of a hodgepodge of things. We're going to revisit the DNA, RNA stuff that we did because neither of you were here last week. And so I want to do another practice one of those and then finish the notes. And then I've got a review sheet for the exam, and we'll go over all that kind of stuff. And then I'll tell you, like, how many multiple choice there are and all that kind of stuff. So we're a little better prepared for this exam than the last one. Okay?
And when is it?
It opens up Sunday.
And then it closes
Tuesday?
Do they let you like go in and look at it and see if there's stuff you want to change on it or anything?
Yep.
They do.
Well, I don't know if they let me, but I can.
It goes this Sunday and it closes Tuesday.
It closes
Wednesday?
Closes Wednesday.
Okay, so for you two that weren't here last week, we talked about protein synthesis, which is not the easiest thing in the world. And if you want me to go back over protein synthesis with you two, I mean, I think we'll have a little bit of time tonight. I'm more than happy to do that. But the biggest thing on the test about, there's a couple of little questions, but one of the bigger things on the test is Here's half of a DNA molecule. What's the other half? So what's the complementary half? And then take this half of the DNA molecule and make what's called a messenger RNA molecule. And that was what we were doing. And I realized some of the recording shut off last week. Sorry about that. If it did, was that on there or not? Was the part about the? I was trying to hold up the paper to show you guys. Was that?
That was Monday,
Yeah.
so I worked until 8
Oh,
o'clock.
so you
last
haven't
night,
looked
so
at
I
it yet.
haven't watched
Shandy,
it.
did, okay, so I know some of it shut off, but we'll walk through that. So, okay, there's two on here. We're gonna do one together, and then I'll let you do the second one on your own. We'll just walk through this first one. And so remember, what are the 4 letters that make up the DNA alphabet? Do you remember?
A, T, G, C.
A, T, C, and G, right. And so we got A, T, C, and G. And then what about the RNA alphabet?
A, T,
A, U, C, and G. Good. Don't speak. You weren't here last week.
I remember this from
I'm
high school.
teasing. I'm just
Actually
teasing
from
you.
middle school.
Ooh, middle school. That's impressive. I'm just
Oh
teasing.
my
you.
gosh, they never taught us this.
Okay. Oh, there's my answer key. Okay, so just look at the one that says it says example number 1. So they've given you half of the DNA molecule. So we've got the first 3 are A, T, and G. So what would the complementary base pairs be to A, T, and G? It would A pairs with
T,
T with
A,
G with
C.
We'll finish that row. That's the easy one.
My teacher's name was GC
Mm-hmm.
for Gordon Coy, and she told us amazing teacher GC, and I literally that's probably the only thing I
Oh,
remember
that's
from
so
8th grade
funny.
Science.
Oh, that is funny.
You got one for the other one?
I just know that it switches out, so the fact that it only did it sometimes made me remember.
Gotcha. Okay, so you want me to go? Are you ready for me to go over the rest of that line? Yes? No? Did somebody say no? No. Everybody said yes. Okay. So it should read from left to right: T A C C A T C G A T T G G A A. Yeah. Yeah. Good. Good. Okay. Now, do you want to make a messenger RNA molecule out of the top row or the row that you just did? Top row? Fine. I can do it either way. I'm just.
You're asking?
I'm asking.
I already
Oh.
started using the bottom row.
Oh, that's good because that's the one I have written down.
Okay.
Okay, so now let's make a messenger RNA from the one you just filled in. So the bottom row. So remember, even though there is no T in RNA, RNA recognizes a T. It says, oh yeah, I know you. I don't have any of you, but I know you. So in this case, what would be the first nitrogen base in that messenger RNA if we are transcribing the bottom row? A. In that case, T would pair with an A. T can read an A, it just, or messenger RNA can read a T, sorry about that, it just doesn't have any. Now, the second one up in the DNA is an A, now what would pair with that?
U.
U. And C still pairs with G. Do you want me to keep going, or you want to do it on your own? Yes, what, yes? Oh, on your own, okay. What'd you say? That's the one I had. That's the one I had written on my answer key. That's what I do in high school. But if you did the top row, did you do the top row already? What'd you have there? Yeah, no, that's fine. I mean, I can.
Oh, no, like I got it. No, I just made like,
It's perfectly right. Yay, cool. Okay. So yours will just.
No,
be
yeah,
different.
no.
But that's okay because I can come back and check. else that had already started transcribing that top row? Everybody else has the bottom row? That's okay, you're special. Okay, are you good? Are you ready or no? Okay, so here's what your messenger RNA should read if you transcribe the bottom row above that. It should read AUG, GUA, GCU, AAC, CUU. Got it right? Any questions on that? Okay, you're good, Sarah. Shandy, are you?
I'm good.
You're good? Okay, now, what is the transfer, so we've got the transfer RNA then. We've got this transfer, and those three nitrogen bases on a messenger RNA, do you remember what I called those? It's not in the, codon. So, yeah, they don't use that term on the exam, but it's just easier for me if I just keep doing it. Okay. And then remember, you've got the transfer RNAs with the anticodon, which is going to be just the matching base pairs. So if you look at the transfer RNA from your messenger RNA, it's just the complementary base pair. This is beyond what you need for the test. So if you want me to do it, I will. If you want me to stop, I will do that as well. I know some people like to see the finished product, some people don't, and I'm okay either way. You guys just tell me. As far as the test goes, is making that messenger RNA, and that's it.
I'm good with that.
You're good with that?
How are we gonna know which strand to go from?
It only gives you one.
Okay.
Yep. The question, yeah, luckily the question only gives you one.
Okay.
It says, the first one, I think, says something to the effect of give the complementary DNA base pairs, and then the second one says something to the effect of give the complementary messenger RNA. So it only gives you one strand, so there will be no confusion. Okay? Just make sure, and there's no multiple choice on that, you've got to write it down. So just make sure when you're doing RNA, look through it when you're all done, make sure there aren't any T's in there. There should be U's and no T's in there, okay? So then I've given you another one at the bottom that you can do for practice, whatever. I can post the results later, or I can give you the results or give you the answer later on if you want, but I just wanted to go back through that. Okay? Are we okay with that? We're good? Fabulous. All right. I'll put that aside. Oh, here. I better show. Just in case you want to see, take a screenshot, and there's the bottom one. Okay. Groovy. All right. So, Monday night, we stopped with, we did osmosis, diffusion, endocytosis, did we do?
Yes.
We did. Didn't we do endocytosis? We did, didn't we? Did I get that far?
I think we're on page 20.
The last thing I wrote was isotonic, hypertonic,
Oh.
hypotonic.
Really?
I thought we did filtration.
Unless I like.
No, I didn't get as far as I thought then. That's fine. That is not a problem.
Because I thought we got as far as try this at home with find the watery fruit.
Oh, we got to this?
Yeah,
Oh.
because you said you can do this at home. And
Oh,
I said,
didn't I tell you about my recipe with my, yeah, I was making fun of my tomato tart. Sounds terrible. Okay. So I did not talk about. Okay, so we didn't get this far. Okay, so, I did talk about diffusion. We talked about diffusion? I talked about osmosis, which we talked about this.
Yeah.
Hypotonic, or hypotonic. Great.
Yeah.
If I put red blood cells into a isotonic solution, will they swell up, shrink, or stay the same? Stay the same. If I put it in a hypotonic solution, will they swell up, stay the same, or shrink? Swell up and then eventually burst, right? And hypertonic, like salt water, what will they do?
shrink, and the term they use is crenation, which is a weird word. Okay, we did that. Stop there. Okay. Oh, I see where we are. Alrighty. Sorry, I lost track. Okay. So, in your body, there are lots of things that you are going to want to filter out. So think about, for instance, your kidneys. Okay, all of the things that you. Darn it! There we go. All the things that you were eating and drinking today is all going through your digestive tract. It's going through your bloodstream. And your blood is going to go through your kidneys. And there's little units in your kidneys called nephrons. And nephrons have these silly little crazy straws attached to them. And in these crazy straws, all of that fluid that's going through there, some of it needs to be filtered out because we want to try to get rid of the waste products. And then some of it is going to be reabsorbed. So filtration happens a lot. It's due to hydrostatic pressure. And I don't think there's a question about that on the test. Mostly this is at capillary walls where we want to get rid of any waste products and put in, keep anything that is good. Happens in your kidneys, happens all over the place. Kidneys is the one that I always think of though, because you're filtering out things. And so, well, no, it's because I forgot I hadn't done this part yet. I was thinking I'd already done this. Just trying to think. Just give me just a second. Okay. Not all substances can just diffuse straight through a cell membrane. Sometimes they need what are called carrier proteins. Carrier proteins are very specific. If I needed help moving through a cell membrane, I was too big, of course I am, to fit through the actual cell membrane, and I needed somebody to carry me over, I would have my own carrier protein. Bree would have her own carrier protein. Brooke would have her own carrier protein. Because these carrier proteins that are going to help specific molecules cross the cell membrane are specific. If I needed help, or I really wanted to get inside of a cell, but there wasn't a carrier protein for me, I wouldn't be allowed in. So this is one way that cells keep things out that they don't want in. Sorry, you can't come in. I don't have a carrier protein for you. If all of the carrier proteins are full, that means that your cell is going to be saturated. So glucose, for instance, if all of the carrier proteins that help glucose across the cell membrane, they're all full, they've all been used, your cells have taken in all the glucose that they can possibly take in, and they would be called saturated. You've got certain hormones that regulate that activity. For instance, if we're talking about glucose, we're talking about insulin, which is a hormone that's going to regulate those proteins that help these different molecules get across the cell membrane. There are two types of this carrier-mediated transport. There's facilitated transport, and then there's active. Remember when we talked about diffusion and osmosis on Monday? Everything was passive. No energy was used. Everything was going from an area of greater concentration to an area of lesser concentration. Oops, got too many computers going here. These carrier proteins, most of them are still passive. Okay, even glucose, those carrier proteins, they're still going from an area of greater concentration to lesser concentration. No energy is being used. They're just too big to fit through the cell membrane. That's called facilitated transport. A molecule that cannot normally pass through a cell membrane can do so passively through a carrier protein. We're still going from an area of greater concentration to an area of lesser concentration, even though they need help. The only time energy is needed to get molecules across the cell membrane, we call it active transport. And that usually only happens in a couple of different times. One is if we're going from a lesser concentration to a greater concentration. It's kind of like salmon swimming upstream. You know how hard that would be? Well, how much energy that would take?
It's the same thing for molecules. So instead of going from greater to lesser, which is easy, it's going down a slide. Active transport is using energy to move molecules in the opposite direction. And they're like sodium potassium pump. Sometimes that happens. Usually this has to do with ions in your body for different physiological functions. You don't need to know what those functions are right now. This is, again, sodium potassium pump. This is a case where energy would need to be used. And that's what this is showing you here. ATP, we're releasing, breaking the bond between the second and third phosphates, releasing energy, so you have ADP left, but it's allowing these molecules to go from a lesser concentration to a greater concentration. That's one of the examples. The other example of when active transport would happen, when energy is needed, is if a cell wants to get in like a really, really big molecule of something. Like the biggest cheeseburger you've ever seen. And I'm like, oh, I really want that cheeseburger. Well, it's going to take some energy to get that in my body. Same thing with the cell. Cells do what's called endocytosis. Endo bringing in cytosis cells. And there's three types of endocytosis. There's receptor-mediated phagocytosis and pinocytosis. So if there is a big piece of food, and I really want that big piece of, sorry, you have to be my piece of food. I really want that piece of food in my cell because I think that would make a great dinner, okay? But it's too big just to diffuse through the cell membrane. It's too big to go through a special protein, but darn it, I am going to bring you in. So literally what happens is the cell membrane makes a divot like this, and then the membrane kind of then surrounds that big food particle and brings it in. Macrophages are your body's Pac-Man. They do this like if she's a really big pathogen, she's going to hurt me. This is what a macrophage would do. I'm going to take you in and I'm going to destroy you.
Hide Shannon.
Thank you.
I don't want you to fall. You're moving too much.
I know. I appreciate that. That's a food particle. That's called phagocytosis. Of course, the teenage boys love that. Oh, phago, you know.
Oh my god.
Yeah, they do. They love that. Now, if instead of a big food particle, there was a large fluid with some solutes in it that a cell wanted to bring in, and again, it would be along those same lines. It could be a big fluid filled with a whole pack of bacteria. Cell's going to do the same thing. A macrophage would do the same thing. But because it's a liquid, it's called pinocytosis. So phagocytosis is bringing in big food particles. Pinocytosis is bringing in big amounts of liquid, big amounts of liquid.
Like 6 ounces of Tito's?
Yes! Woohoo!
That was me last night.
Okay, so what's this receptor-mediated vesicle thing? Do you guys know anything? I don't know what your background is. Do you know anything about neurotransmitters and the nervous system and
I don't.
yeah, okay. So let me give you a quick lesson on neurons and neurotransmitters. So let's say that over against this window, there is a neuron. And over against that wall, there's another neuron. And I need to get a message from that neuron to this neuron. Well, the message itself can't just hop. Where you are sitting, it's called a cleft. It's like the Grand Canyon of your cells, okay? Chemical signals need to bring that message over because the message itself can't cross it. So it's got to be chemical signals. So those neurotransmitters are the chemicals that are going to bring that across. So over here, there would be receptacles. And these receptacles would literally receive the neurotransmitters. That's what receptor-mediated endocytosis is. It is just a special type of getting larger molecules from point A to point B. Specifically, that one is going to take, honestly, I don't know where else in the body that takes place,
except for where you're talking about neurotransmitters, like acetylcholine, dopamine, serotonin, all those kinds of things. So it's just a third type of this big transport. So these three bring things into the cell. Well, we all know that eventually there's going to be some waste products. Well, your cells don't want to keep the waste products out. So exocytosis is getting rid of the waste products that were brought in through endocytosis. And literally, this big vacuole, what it does is it moves to the cell membrane, merges with the cell membrane, and then spits out the waste products. I don't know if there's any pictures on this or not. These are vesicles. Okay. So they're bringing in. Oh, this is a good part right there. So like this would be bringing in fluids from the outside and making vesicles. There's phagocytosis. There's a big old bacteria being brought in to a macrophage, surrounded and then getting destroyed, and then we're spitting out the excess stuff. Now that takes energy, okay? That's active transport. Pretty much everything else we talked about on Monday, that's all passive, no energy. Okay, good, good. Moving on. That's just more of it. Okay, wait a minute. All right, we are ready to finish up. We're going to talk about cell division. First of all, what is a somatic cell? Do you guys know what soma means? Soma means body. So somatic is a body cell. So muscle, skin, bone, anything like that. Those are somatic cells. Your body, you've got two main types of cells. You have body cells, you have sex cells. And of course, sex cells are eggs and sperm. Everything else is a somatic cell or a body cell. Do all cells have the ability to divide and regenerate? Yes or no? I got one of each.
Divide into two?
Divide and regenerate. No, because what can't regenerate? Not my hair. My hair regenerates big time. Nervous tissue. Nervous tissue. Think about that. Nerve cells cannot. Think about how great it would be if nerve cells could regenerate. Anybody that had a spinal cord injury, they'd be able to walk again. So nervous tissue cannot regenerate. Most of the cells in your body can, but they can't all regenerate.
That's kind of the same with the heart and the muscles. They can to an extent.
Yes, to an extent. Technically, what's interesting is your textbook will tell you muscles cannot, which I found a little bit interesting. But I think part of that is because, like, if you tear a muscle, when it heals, you form a lot of scar tissue in there. So I think that's what they're taking into account. You're not really making new muscle cells. They're not dividing. Yeah. So that's, yeah. You're sticking a Band-Aid on, exactly. And what do centrioles do? Do you remember those from when we did organelles?
Those are just little things like this. No.
I don't know. That was phospholipids. That was phospholipids.
No, those
Centrioles
are
are-
microvilli.
Centrioles. You guys are going to get some other professor someday, and you're going to do these weird things. I'm like, who the hell taught you
These are
that?
the microvilli.
Spirit fingers. No, these are not spirit fingers. Centrioles are in animal cells that can divide. So centrioles are not in plant cells because plant cells divide differently than animal cells. So centrioles we're going to talk about now that we're talking about mitosis. Okay?
That can divide.
That can divide. So you wouldn't find centrioles in nervous tissue. You actually won't find centrioles in muscle tissue. But in your skin and bones and having a brain fart right now, any other tissues in your body, you would find them. Okay, moving on. Good. Oopsie. There we go. Do you guys know much at all about mitosis and meiosis or a cell cycle? Yeah, no, not really. I'll just start at the get-go. Okay. Do you recognize the terms mitosis and meiosis, or at least one of them? All right. So, life cycle of a cell, the whole thing is we need to take one cell and get two cells. And then you take two cells and you get four, and four and you get eight, so on and so forth. Mitosis is what we call the process to make body cells or somatic cells. Meiosis is the name of the process to get sex cells. And this chapter does not discuss meiosis. We're only going to talk about mitosis. Body cells, not sex cells. What? Well, okay, let me I'm going to give you kind of an overview of what happens here. So we're going to be talking about the quote-unquote life cycle of a cell. All right, now cells have a job to do, but they only do that job for a short period of, well, no, I shouldn't say short, for a certain period of time, because every type of cell will be different. The other parts of the time, they're either dividing or they're getting ready to divide. So we call this whole thing the cell cycle. The whole thing isn't mitosis. The whole thing is referred to as the cell cycle. So let's say we have a brand new cell, just got done going through mitosis. It's going to go through 3 to 4 stages, it depends, of something called interphase. Here's a little way for you to remember what we're going to talk about today. IPMAT-C, and I'll explain as I go. This is a little thing that we use in high school for the kids to remember what all happens during a cell cycle. IPMAT. See? That's usually how I go. IPMAT. See? I stands for, and you don't have to write this down quite yet because this you can't, but we'll get to it on the next few slides. But this I stands for interphase. And there are 4 different parts of interphase. And most cells are in interphase for a really long time. We actually do a lab where we look at onion root tips. And the kids can figure out how many hours an onion spends in interphase, and then how short is metaphase, and things like that. So we'll go through what happens in each of these. Interphase truly isn't talked about much in your textbook, okay? But interphase, this is right after the cell is made. Some cells spend a little bit of time in, this is called G0. Some cells don't spend any time here at all because they got things to do. They're busy. I always tell the kids G1 means grow. You know, when a new cell is made, it's little. Just like a new baby is made, it's little. So it needs to get bigger. It needs to make some organelles that it doesn't have. That's what is going to be in G1, and this is all part of interphase. S stands for synthesis. That's when DNA replication takes place. Remember, we haven't talked really about that yet. G2, I always call this the getting ready stage, because at this point the cell's getting ready to divide. So this G0, G1, S, and G2, all of that makes up interphase, which is part of the cell cycle but not part of mitosis. People get that confused all the time. Mitosis is PMAT. Prophase, metaphase, anaphase, telophase. And then cytokinesis, which is sort of kind of part of mitosis, but not really. So that's an overview of where we're going, okay? 4 phases of mitosis, PMAT. IPMAT-C is the whole cell cycle, the whole life cycle of a cell. Just mitosis, just the dividing part, is PMAT. We'll talk about what the C stands for in a couple minutes. Again, more pictures. We'll get to the pictures in a minute. I'm going to talk about the phases, and then I'll come back and show you with the pictures what's happening.
Phase 1 is called prophase. That's the P, prophase. Up until this point, you couldn't actually see chromosomes. So during prophase, the chromosomes become visible. Do you remember, was it this class? Did I talk about how, I think I did. Chromosomes most of the time are like an unraveled piece of yarn, and then they condense to look like chromosomes. Okay, so up until this point, you can't see the chromosomes because they're the unraveled piece of yarn. But now that yarn is going to condense, and you can see the chromosomes. Chromosomes actually are in two parts. We refer to those as chromatids, or sometimes you're going to call them sister chromatids. So, you know, we use kind of a representation of a letter X a lot of times to represent what a chromosome looks like. Well, it's actually two parts. This is one chromatid, and that's the other chromatid. So sometimes we call them sister chromatids. The whole thing together is a chromosome. Again, not different than anything else we talked about, but science loves to give things different names. They're held together by a scrunchie. And the scrunchie is called a centromere. So now, here's where those centrioles come in, and this is all still prophase. During prophase, then, those centrioles are going to separate. And one's going to go to one end of the cell, and the other's going to go to the other end of the cell. I think of those like clotheslines. So if you ever had a clothesline in your backyard and you got a wooden pole here and a wooden pole here, and you got the line between them to hang your clothes on, well, the poles are the chromatids. The line are these things called spindle fibers. Spindle fibers are made of microtubules, which we talked about earlier. Oh my gosh. Okay, so this is prophase. This is early. Now, prophase takes a long time because a lot of things are going on. So usually we talk about early prophase, sometimes talk about middle prophase, sometimes talk about late prophase. So if you ever see pictures, usually prophase has several different pictures. This is early prophase. Remember how I said the chromosomes are going to condense to form, or the chromatid is going to condense to form chromosomes. This right there is still chromatin, hasn't condensed yet. There's the centrioles separating, and they end up going one centriole to one end of the cell, one centriole to the other. We've now got our condensed chromosomes. They're hooked on the clothesline, onto the spindle fibers with this centromere. The centromere is literally holding them on. Okay? That's all prophase. Lots of things go on there. Questions on that one? Next one's easy. M is for metaphase. M is also for middle. During metaphase, those chromosomes line up in the middle of the cell. Your book calls it the central zone. The test calls it the central zone. I just always call it the middle because middle and meta both begin with M. And that is literally the only thing that happens during metaphase. Chromosomes line up in the middle, the central zone. That's it for metaphase. The next phase is anaphase. I always call anaphase the traveling phase because at this point those sister chromatids separate. One goes to one end of the cell, one goes to the other end of the cell. So the scrunchie kind of disintegrates and the sister chromatids separate. One goes to one end, one goes to the other end. Yeah, there we go. The sisterhood of the traveling chromatids. But like I call it the traveling phase.
And that's what's happening in this picture. You can see that they have split apart. One goes to one end, one goes to the other end. At this point, it almost immediately moves into the last phase, which is telophase, and it says cleavage starts. Cleaving is just when the two cells start to pinch in half. So it starts to cleave at the end of anaphase, but really the complete cleavage takes place during telophase. Telophase is the last phase. During telophase, cytokinesis also happens. So I always say telophase and cytokinesis happen at the same time. Cytokinesis literally just means that the cells completely break in half. So if you look here, this is telophase. You can see that the cells are starting to break in half. Cytokinesis is when they totally break in half. Now, depending on textbooks, depending on professors, sometimes you'll say telophase and cytokinesis together. Sometimes we say that cytokinesis is part of the whole entire cell cycle, but not part of mitosis. Unfortunately, it depends on who's teaching it and what textbook they're using. It is always safe to know, though, that PMAT is mitosis. Prophase, metaphase, anaphase, telophase is mitosis. Cytokinesis usually comes after, even though it happens at the same time as telophase. It's kind of one of those gray areas when you're teaching. So IPMAT-C is the entire cell cycle. PMAT is just mitosis, cell division. Does that make sense? But Rachel, question?
No.
Oh.
Sorry.
It's all right.
It's the brat.
Oh, she's not a brat. Okay. I love this. Help. So here are resources if you need them. I do have a short, speaking of resources, I do have a short mitosis video I want to show you. Since we just talked about all this, hopefully the visual will help a little bit. And then, oh yeah, we're in good shape. And then we've got about a half an hour to start reviewing, okay? It is a 6-minute video.
Estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day, our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell.
This is the
DNA
unraveled yarn.
condenses by wrapping
Okay,
around cores
that's
of
when it
histone
condenses.
proteins, forming nucleosomes. This beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into 3 distinct phases. During G1
most videos
or
don't
Gap
use
1
G0.
phase, all the organelles
And you don't need to know much
cytoplasmic
about this anyway.
components, including the centrioles in animal cells, replicate. Then during S, or synthesis phase, the DNA replicates. Finally, during G2, or Gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase,
PMAT.
anaphase, and telophase. prophase, the chromosomes condense and become visible, appearing as two sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus.
I
And
call
the
it spindle
nuclear
fibers.
envelope breaks down. Towards the end of prophase, chromosomes attach by proteins in their centromeres called kinetochores to microtubules from each pole, moving the chromosomes toward the equator of the cell.
Middle.
During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual
Yes, the
chromosomes.
scrunchie breaks down.
The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell.
Looks like
This
boobs.
indentation is
It
made
does.
from a constricting box of actin
Her
filaments
nipples are sticking
surrounding
the
the
wrong way, but.
inside of the cell's circumference.
Woo! Cleavage phase.
Chromosomes cluster at opposite poles
Ooh, it's a
and
cluster.
begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form
There's
the
a lot of this detail
cytoskeleton
you don't
of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately.
That's good enough. But that at least gives you a visual of everything we talked about. Okay, good? Questions on that? All right, let's talk about the exam. You're welcome. It's front and back.
Can you just specify what's a chromatin? What's the chromatin?
Chromatin, so the chromosome is when it's condensed and looks like a chromosome. When it unravels, it's just called chromatin. It's just another name for the same stuff. Yeah, it's not, it can be very confusing because we use several names for the same thing. Bitte?
What
Oh.
did you say?
Bitte, German for you're welcome. I know very little German. I took German in high school, and the teacher only stayed for 2 years so we could get our second year in. So I learned really weird things like ich habe ein Wassermelone, which means I have a watermelon. Rudi hiemt for Freude, which means Rudy jumped for joy. And ich bin kalt, which we all thought meant I am cold, but apparently it means I'm sexually frigid. And even if she just told us that to get a rise out of us, it's still pretty entertaining. So I use that periodically. I just throw that out at parties and things because. Okay. So I'll post a copy of this in Blackboard. Okay, so what I did was I went through question by question to kind of to look for specific things that you were going to need. They're not necessarily in this order because, for instance, if you look at like under pH, there's 5 different things. So they were probably questions that were kind of scattered around. But I lumped them all together because they're pH. But I can tell you, cellular respiration is the first question. So we talked about cellular respiration Monday or last week? Last week, okay. And remember, cellular respiration is what takes place it would have been Monday because we talked about mitochondria Monday, right? Whatever. Cellular respiration takes place in the mitochondria. And so, if you remember, when we talked about cellular respiration, we had
here's the mitochondria. It's got that real funny thing inside. And then outside of the mitochondria is where the glucose is, C6H12O6. Outside of here is the very first stage of when this breaks down to make ATP. And the process that breaks that down is called glycolysis. Oh, I can't spell. That is not right, I don't think. Anyway, all that means is a molecule of glucose gets broken down into, do you remember what two somethings? Do you remember what that is? Two molecules of pyruvate or pyruvic acid. Sometimes it's called pyruvate, sometimes it's called pyruvic acid, so don't let that mess you up depending on what class you take. It's the same thing. And then if there's oxygen, this pyruvic acid goes into the mitochondria and the rest of cellular respiration takes place to get ATP. If there isn't any oxygen, do you remember what forms in our muscles? Lactic acid, right. And if we're making beer and wine, it's alcohol. Yeah, without oxygen.
So that's what happens like when runners
Yep.
after they run so long they lift their legs up.
So they've used up all the oxygen in their muscle tissue, but they're not going to just fall over in the middle of a race. Their body's going to keep going, but it's got to produce ATP without any oxygen left in their muscles. So it's producing it anaerobically, and a byproduct of that is lactic acid. And so when they're all done with the race, if they don't try to get rid of the lactic acid, that's when they cramp up. So that's why if you ever notice runners, they're not supposed to just, you come across the finish line and they just die. They just stop. If you ever notice, runners keep jogging and breathing slowly, but that's to replenish the oxygen. Sometimes I've seen them lay down, put their legs up to drain the lactic acid. I'm not sure I buy that, but I've seen them do that too. Okay? All right, so that is, I will double check on why I wrote down ATP production. I think I will double check on why I wrote that one down there, but I will do that when I can use this computer. pH, there are several different questions about pH. Now, we talked briefly about the fact that pH is logarithmic, and if a pH of 6 is 10 times more acidic than a pH of 7, a pH of 5 is 100 times more acidic than a pH of 7, 4 is 1,000 times more acidic. I can tell you that there's a question like that on there with different numbers. So it's not 6, 5, and 4, but just look at the pattern. Okay? So if the pH is just 1 down from what they're asking, it's 10 times. If it's 2 down from what they're asking, it's 100 times. And if it's 3 down from what they're asking, it's 1,000 times. See what I'm saying? Yeah, that's just a pattern. Strong acid and strong bases. So acids have more hydrogen ions and bases have more hydroxide ions. Hydrogen ion is H+, hydroxide is OH negative. Sometimes they say acids have more hydrogen ions, bases have less hydrogen ions. They just switch up the wording. Instead of throwing the hydroxide ions in there, they only talk about hydrogen ions. So if that's the case, acids have more hydrogen ions, bases have less hydrogen ions. So a stronger acid would have more hydrogen ions. 1 is the strongest acid you can have. 14 is the strongest base you can have. Stronger base would have less hydrogen ions, more hydroxide ions. And what exactly does pH measure?
I feel like this is a trick question.
That's what everybody says. It measures acidity. What does that mean?
In your urine.
What does pH actually measure? And I know, it's fine, because unless you specifically spelled this out, everybody misses that question. pH measures the amount of hydrogen ions in a solution. Is that what you're going to say?
Yes, I swear.
Sure, Sierra, sure it was. It measures the amount of hydrogen ions in a solution. That is what that does. pH, the P stands for, shoot. It's not percentage, it is power of hydrogen. Yeah, power of hydrogen. Okay? There is a question about, so when we talk about ATP, we usually talk about carbohydrates, just getting energy in the form of ATP out of carbohydrates. Is that the only macromolecule you can get energy from? Can you get energy from lipids? Can you get energy from proteins? Sure. So it's a weird question, but just know that it's, yeah, you can get ATP out of things other than just carbohydrates. And AMP, so remember how I had what?
Monophilic, is it monophilic? Is that right?
I think you're right. No? I remember asking this, but I don't
So
remember.
ATP, adenosine triphosphates, means there's 3 phosphate molecules. ADP means there's only 2 phosphates. AMP means there's only 1 phosphate. Do you remember the term monomer?
Yeah.
A monomer makes up a polymer. So if you were asked what would the monomer of ATP be, it's AMP. What's the small unit that would go together to make a large unit of ATP? It's AMP. I've never used AMP as a monomer before, but you will this week.
So does the M in that stand for mono?
Mono.
Okay. Or
Stands for mono.
Di,
Adenosine,
tri,
yeah.
or one,
Monoditride. I'm being Vanna. Hmm? Yes. You need to know the definition of I'm going to speed up just a little bit. Some of this you can, the things that I know you can find on your own, because it's 7:28 already, so I want to speed up just a titch. Make sure you know the definition of metabolism. Macromolecules, remember, are also called organic compounds because they all have carbon in them. The 4 macromolecules, remember what they are? Carbohydrates, proteins, lipids, nucleic acids. Make sure you know what monomer makes up all of those. So the monomer for a carbohydrate is a monosaccharide. The monomer for a protein is amino acid. The monomer for a lipid is fatty acid and glycerol. The monomer for nucleic acid is nucleotide. So make sure you compare those up. Okay? It's either a matching or it's a multiple choice, like which one is wrong. Can't remember exactly which question that was, but you need to know the pairs. Glycogen. Do you guys remember what glycogen is? Glycogen is how animals store carbohydrates. So glycogen is a carbohydrate. Just FYI. Look up, make sure you know examples of lipids. What are some of the lipids we talked about? What are some of the monosaccharides that we talked about? Glucose, for instance, is a monosaccharide. Lipids are, steroids are lipids. Okay, you might want to know that. Oils, fats, waxes. Mitosis we just talked about, so this should be fresh. This is why that one question was on the first slide. Do all body cells, soma cells, divide by mitosis? We answered that already. Know the stages, PMAT, what cytokinesis, know what interphase is, and then I just put IPMAT-C so that you would remember that all of those together make up the cell cycle. Protein synthesis we did Monday. Know that, remember that transfer RNA and anticodon go together. Okay, transfer RNA and anticodon go together. So, and the transfer RNA carries an amino acid. So, bless you.
When the ribosome reads the messenger RNA, it calls for the transfer RNA with the correct amino acid and the correct transfer RNA on it. So just remember that those things all go together. B and C we've done. Complete a DNA molecule, make messenger RNA molecule. When we are making a protein, I think I talked about this, but I can't remember what chapter I talked about it in. When we make a protein, at first, when protein synthesis is over, it's just a long chain of amino acids. That's a primary structure, okay? A long chain of amino acids is a primary structure. A long chain of amino acids is a primary structure. A primary structure is a long chain of amino acids. And then it'll go through, it'll fold a little bit, it'll become a secondary. It'll fold a little bit more, it'll become a tertiary, and then it'll fold finally and become quaternary, which I have a hard time saying that word. But that's when it is finally called a protein, after it completely folds up. What is made during transcription? So remember, transcription takes place in the nucleus. So what kind of RNA is made in the nucleus during transcription? What was the letter we put in front of it? M. Messenger RNA is made during transcription. Know the kind of bond that forms between amino acids, and know that a polypeptide is a chain of amino acids. Then we get into chemistry. I know, everybody's least favorite. Isotopes, atomic mass, and atomic number. Make sure you know those. So like if I give you carbon and I say the atomic number is 6 and the atomic mass is 12. The atomic number tells you how many what that it has.
Electrons.
And also protons. Yeah, because those all is equal. So then atomic mass is what two things added together?
The protons and then
Right, protons and neutrons. So what if one atom of carbon had a certain number of neutrons, but another atom of carbon has a different number of neutrons? What would we refer to those as? Isotopes. Yep, isotopes. Denaturation, atomic mass, sorry, wait. Denaturation is a breakdown of proteins. What is hydrostatic pressure? I forget what, give me a second, because I forget why I wrote that one down. It'll come back to me. Covalent versus ionic bonds. Which ones share?
covalent.
And what do ionic bonds do? They take away or steal. Which ones are stronger? Covalent. Do things formed by covalent bonds, can you break those down easily in water? No. Think about ionic bond, think about salt. When you have a sore throat, do you ever take salt, put it in water, stir it around and gargle? Does it dissolve easily? Sure. That's an ionic bond, will dissolve easily. Covalent bond will not. What makes an atom stable? If I want helium, for instance. Helium has 2 electrons in its outer energy level, but it only has one energy level. Why would I consider that stable? It's full. Yeah, it's full. Anytime an outer energy level is full, it's stable. Triglycerides. Oh, sorry, I skipped one. My bad. What all do enzymes do? Let's put it this way. They do a lot of things. Enzymes do a lot of things. Like on a multiple choice question where pick all that apply, they do a lot of things. So make sure you look up what all enzymes do. They do a lot of things. A triglyceride is the best way to store, sorry, brain fart. Triglyceride is your body's best way to store fats as a triglyceride. Dehydration synthesis, I have to come back to that one. The sodium potassium pump, there is a question. So remember we were talking about that and Rachel said there was when she was watching the video, there was something on the video about 3 sodium out, 2 potassium in, or whatever that was. 2 and 3, yeah. And I made sure everybody circled it or wrote it down or something. Look at what that exchange is. Like, I can't remember what it is off the top of my head, but that's on there. Know the difference between catabolism and anabolism. An example of what that would be. I want to go just a little, I got to look up a couple things here for you real quick. Know the difference between microvilli and cilia. Which one increase surface area for absorption? Microvilli. And what does cilia do? Move. Difference between active and passive transport. That's what we just did here. It is a matching. You have to, there's a whole list of things, and you have to match which one's active, which one's passive. Honestly, if you just remember which ones are active, everything else is passive. So active is pinocytosis, phagocytosis, the vesicle. Shoot. What was the one with the neurotransmitters? I'm going too fast, so my brain is not keep. You know what I'm talking about, though, right? On that slide. And exocytosis. Those are active. So passive is everything else. Just make sure. Thank you. Receptor mediated. Thank you. So make sure you can pair those up. Know the organelles and their functions. That's a matching section as well. Hydrophilic and hydrophobic for the cell membrane. We've done that a gazillion times. Make sure you know which is which. What all do proteins in the cell membrane do? They do a lot as well. I mean, a lot. Choose all that apply. A lot. So make sure you look at all the things that proteins do. Know the difference between diffusion and osmosis. Diffusion is the movement of solutes from a greater to a lesser concentration. Osmosis is the movement of water. Why does filtration occur? That was, I think, like the first slide today. Hydrostatic pressure. Know the characteristics of water, like it's a good solvent. I can't remember what else is on there. I'll look here in a second. And then glycocalyx, we talked about in this last chapter. I wonder if I, never mind, I can pull it up. Hold on. Look at all these things that proteins do in the cell membrane. Just look at them all. There's 6 of them. Just FYI. There's glycocalyx. Okay. So this is, there it is. Functions of glycocalyx. Make sure you look these over. Again, it's an all, choose all that apply. So that is in this chapter's notes. By the way, cytosol and cytoplasm are the same thing, okay? Cytosol and cytoplasm are the same thing. Sorry, I gotta shut down.